Hello and welcome to our webinar today. We really appreciate you joining us for today's topic of Organ on Chip 101, your introduction to the future of biomedical research. Before we start, I just would like to do a bit of housekeeping. So if you look on the left-hand side, in this app, you see a control panel and there is a chat section. So this one you can use for if there's like any general requests, right? Like you can't hear something or can I get the recording, that sort of thing. And there's also a Q&A section, and I would ask you if you've got any questions to us regarding the content of this webinar, so any sort of research questions, then please use that one. That'd be really great. And then otherwise, we will be sending the recording to you afterwards. It will be sometime around next week, but we'll definitely come, and you can rewatch it or show it to your colleagues. Okay, so before I will introduce our speakers, I've got a little poll because I would like to hear how you heard about us, how you found us online. So while you're going to answer this, I'm going to introduce our speakers for today. So first up, we will have Marty Steiger. He's an application specialist at Dynamic 42, and he helps us he helps scientists and our industry partners to leverage organ-on-chip technology, and his background spans bioprinting, comparative and aging biology, and he's worked on building advanced 3D in vitro models to improve research and therapeutic discovery. He will be followed by Tim Carden, who's recently handed in his doctoral thesis and has now transitioned into an application specialist role at Dynamic 42. He studied molecular medicine and earned his degree from the Friedrich Schiller University here in Jena, where we're based. And with over 6 years of hands-on experience of organ-on-chip systems, he's focused on liver and intestine models. His work included investigating drug-induced liver injury in liver models to better understand liver toxicity. And more recently, his research has concentrated on intestinal host microbiota interactions in the context of inflammatory bowel disease, exploring novel therapeutic strategies to modulate these outcomes. That was quite a mouthful. So now that you've learned a bit more about our two speakers, we would like to learn about you. So on my next poll is going to be, what's your application and model of interest? That's always really interesting to us, specifically if we look at like publishing more webinars. Okay, so now without further ado, I would like to hand over to Marty.
Thanks very much for joining us. And I'm just today we're going to talk a little bit more about giving you an introduction into organ-on-chip technology. To start with, just introducing the company as well. So we're located in central Thuringia, and we've been up and running since 2018. So initially, we're going to talk a little bit about why move beyond traditional models, what are the core features of organ-on-chip technology, briefly touch on systems overview and materials that constitute this technology, and then Tim's going to walk you through some model applications that we regularly work with here at Dynamic 42, and some exciting stuff that we've got coming up in the very near future, along with answering your questions at the end. So if we look at the history of cell culture over the past century, what we can see is that model complexity has gradually increased, and it's also increased concomitant with the knowledge that we've basically developed. So as with all things human, the more we learn there is to learn. And we also develop new tools, and they allow us to ask increasingly complex questions. And we also need increasingly complex models to ask those questions as well. In addition to that, if we look at from a translational point of view, we've seen that the existing preclinical models have not worked so well up to date. So currently we only have around 6% of drugs entering phase 1 trials that actually get through to approval from preclinical. So phase 1 is obviously where we're screening for safety and toxicity, and phase 2 is where we start to look for efficacy. And we can see that the vast majority of failures occur in phase 1 and phase 2. And this implicates preclinical models as potentially being a cause for this. Now, of course, it's multifactorial, increasing regulatory burdens. Obviously, it's drugs are becoming more and more targeted as well. But preclinical models are definitely something that we need to sort of look at improving. And we can see that the governments around the world, so the FDA in the US and the EMA here in Europe, they've also started to sort of implement regulatory frameworks to address some of these issues. FDA Modernization Act 2.0 removed blanket animal mandates and recognized the utility of non-animal models. And more recently, the FDA Modernization Act 3.0 really opened up qualification pathways and faster adoption of these models as well. So in regards to the models that we're using, it's essentially a question of choosing the right model for the right question. But these models exist on a continuum. So we have 2D, which is high control and high throughput, but relatively low physiological context on one end. And we have the animal models on the other end, where we get systemic, low throughputs, so a lot of complex physiological data, but they're constrained by the interspecies variations that you get as well. So generally speaking, what we want to do is choose the right model for the right model, and also sort of triage, so using the simplest model that answers the experimental question that you may have for a given experiment. These are the legacy models that we have currently. So if we look sort of from left to right, we have our 2D models. So 2D models, relatively cheap, well established. You can do high throughput as well, but there's very little sort of cell-to-cell communication in a tissue context. And in addition to that as well, the cost is low. You can run high throughput as well, but you don't get that sort of physiologically relevant data. Then we develop spheroids and organoids as well. And in this instance, we get more of the sort of structure and the tissue context, but these are relatively static cultures as well. And of course, we touch on animal models. So animal models, high degree of physiological complexity, but we have issues in regards to interspecies difference. There's also the ethical concerns and high costs. And this is where we feel organ-on-a-chip can really step in as a technology which addresses some of these concerns. So we can have more control over our experiments with organ-on-a-chip. We can have the physiological complexity as well. And we can also introduce mechanical cues, which are more analogous to the in vivo situation. So in regards to what we can simulate in the organ-on-chip context, we have the 3D architecture that we can introduce. And we can also build this up in an engineered way to give us this sort of cell-to-cell signaling similar to what we get inside the body. We can also engineer or introduce dynamic cues, so stretch and strain, which mimics vascularization in the body. And we also can have these sort of gradients of oxygen and nutrients through the tissue as you would in the body as well. And also for biological context, you can incorporate resident circulating immune cells, and you can model infection as well. So you can introduce either pathogenic microbiota, or you can introduce sort of commensal bacteria or fungus as well. Just a brief overview of our particular organ-on-a-chip. So this is the basis. This is basically what our organ-on-a-chip technology looks like. So importantly, the material is biocompatible, which is of course very important because you don't want to be putting your cells on a chip where the materials is going to kill the cells. We also use a special material. There's a very low sorbent, which means that you can dose at physiologically relevant levels, which is critical as well. Some of the other organ-on-chip technologies are made from PDMS, for example, material which absorbs a lot of small molecules. In our system, we also have designed it to be very user-friendly. So it's something you can implement into existing workflows. We also have a plug-and-play assembly, so you can basically adapt a little lure plug on top of these little chimneys, which means you can use it with existing peristaltic pumps or syringe pumps as well. Very reasonably priced. And we've developed it in a microscope slide format, which means it's compatible with live imaging. If we take a cross-section through, essentially this is what the chip looks like. So you have the apical section, you have a porous membrane. On top of that, you have an apical side of the membrane, a basal side. You can seed cells on the top and on the bottom. Typically, we'll seed our endothelial cells on the top with a resident or circulating immune cells, and then an epithelial cell on the bottom, which can perform as a barrier. And we can perfuse both of these channels independently. This is what the perfusion setup looks like. So here we've hooked it up to a peristaltic pump, and you can have linear or circular perfusion. We have tubing and reservoirs, which is good if you're doing drug testing or if you don't want to have to change media on a regular basis as well. And this allows for continuous nutrient and oxygen supply, and also independent perfusion of the channels. Importantly, with our system, you can basically, anything that you already do in your workflows with 2D cell culture, you can already use these same assays. So you can do supernatant sampling for your clinical parameters. We're also able to, for example, remove the tissue after you're finished as an endpoint. So you can do immunofluorescence or RNA-seq. We also can do live cell imaging on our chips as well, in case you want to stain and look for example, for mitochondrial activity or immune cell perfusion. And in addition to that as well, you can measure enzymatic activity. These are the models that we regularly use in-house. So we use these for our CRO services, and we can provide full protocols and application support for those models. But importantly, we also support custom organ development, and a lot of our end users, they use the chips to sort of model tissues and organs which are not represented here as well. For us, we basically function as a full service CRO, but in addition, we're able to sell the chips and the kits and basically everything you need to get started if you want to do this in your own lab as well. Coming very soon, we're about to put on the market a kit that enables you to incorporate a biopolymer, so you can have a tumor microenvironment incorporated into the chip as well. We're also developing a tier system, an O2 system, and this is something that Tim will talk a little bit about later. And with that, I'll hand over to Tim.
Thank you very much, Marty, for the nice introduction. Also, warm welcome from my side to the audience. My name is Tim Carden, as already introduced. I'm now a field application specialist at Dynamic 42 with a lot of hands-on experience with the organ-on-chip technology, focusing on liver and intestinal models. And now I would like to invite you to join me the journey to some real-world applications. I prepared 3 real-world applications that we're actually developing and using here at Dynamic 42, and I will also give you a short perspective of what we're currently developing and what you can expect in the next weeks, months, and years from us. So the first application is really a hot topic since the corona pandemic. So modeling microbial infections such as bacteria, viruses, and fungal species is really a huge topic nowadays, and we can mimic this by including these pathogens, also probiotics, for example, for disease treatment into our intestine and lung systems. And I would now like to give you a broad overview about how our lung-on-chip and gut-on-chip models are composed. So on the left-hand side of the slide, you can see that the lung-on-chip, so if you would do a model scheme of the chip model itself, or the biological chip model itself, you would see that, as Marty introduced already, it's a 2-channel system within an interspan membrane. And on top, we integrate our pulmonary epithelial cells, so either cells from the alveolus or the bronchus. So we both develop primary models for the alveolus and the bronchus. And we also integrate the macrophages that are present in this epithelium. And on the other side, we are able to integrate pulmonary endothelial cells mimicking the adjacent vasculature. And we can perfuse the model, so we can not only perfuse cell culture medium inside this, but we can also put on an air liquid interface within the lung-on-chip and perfuse air within this artificial lung. On the other hand side, we have developed the gut-on-chip, which I was working a lot of in the past years during my PhD. So this model consists of intestinal epithelial cells in the bottom channel, and on the other side, in the opposing top channel, we put in endothelial cells and also integrated macrophages. So all our models here at Dynamic 42 are immunocompetent, at least putting in one cell type of immune cells within the system. All right. Then in 2020, we already published the bacterial and viral co-infection model using the lung-on-chip model. So you can see on the left-hand side how the model is composed. And we exposed the model at air-liquid interface to viral and bacterial particles, namely influenza A virus and Staphylococcus aureus. And we wanted to see whether these individual strains are differing in the infection in comparison to a co-infection of these two pathogens. And what we were able to demonstrate is that, especially in the vasculature, we can see that the co-infection of these two pathogens, they really lead to disruption of the vascular barrier integrity. And you can see on the right hand side some immunofluorescent images here shown with E-cadherin staining, which stains the cell-cell contact of the endothelial cells. So the pathogen is really destroying not only the epithelial side, but also the endothelial side. And the co-infection then leads to a breakdown of this endothelial cell barrier. And we were also able to quantify this. So to quantify the damage of the barrier, you usually would do a Fitz-Dextrin assay, which is a barrier permeability assay where you put a dye on the epithelial side and let it diffuse through the barrier. And the more leaky the barrier is, the more of the dye you can retract from the other side of the channel within the chip. And you can see that the co-infection induced the highest barrier permeability within the model. Another showcase is also fungal infection, and we used basically the same alveolus on-chip model that I just presented for the co-infection, and we inoculated, or Dr. Mai Huang basically did this. We inoculated Aspergillus fumigatus, which is known to induce invasive aspergillosis in the lung. We put this in the epithelial compartment, and what this fungus is basically doing, it's attaching to the epithelial side of the tissue, and then it germinates some hyphal structures. So hyphae structures are building up from the cells that you put in. And these hyphae structures are invasive, so they're damaging both the epithelial and the endothelial cell side, and also are able to translocate across the tissue. And this translocation can be quantified. So we've seen in the upper figure shown on the right side, we can see that the number of conidia of Aspergillus are not restricted to the alveolar side, but can also be found after infection in the vascular side. So we were really able to quantify this translocation process. And in the lower image, you can see that the macrophages in our system, they're really the main mediators of the pro-inflammatory reaction upon the pathogen exposure. So we can see on the right-hand side that the levels of IL-1 beta only increase if the macrophages are present in this model. And then a next step further is not only to model the infection in the lung-on-chip model, but also use it as a treatment platform. So we put in a known antifungal drug treatment that is effective against a broad variety of different fungal pathogens, for example, caspofungin in this case. And we put in caspofungin at concentrations that were also clinically relevant, and we can see a dose-dependent decrease of the fungal load and also the invasive hyphae that were forming upon the infection if we're putting in the caspofungin. And a really cool feature of the whole lung-on-chip model is that you can put the caspofungin or any other kind of antifungal treatment within the vascular perfusion, mimicking also the intravenous route of a drug that you would administer in a human patient. So this is really reflecting also the in vivo situation. And also the images in the lower left corner, you can see also that the fungus, which is usually residing in the epithelial side and translocates upon infection in the endothelial side, is restricted only to the epithelial side if you increase the caspofungin concentrations, because the fungus is directly in contact with the drug, which is vascularly perfused, so limiting the translocation of the invasive hyphae. Another fungal infection model can be also seen in the gut-on-chip model. So this is something I recently worked on and was published last year already. So we're using the gut-on-chip model, and here in the chip you can see these little dots here, and these are microcolonies of Candida albicans. And Candida albicans is usually a commensal fungus residing in the gut without causing any harm. But under some predisposing factors, such as immunodepression and other factors, it can become a pathogen. And this is something we mimicked here in the chip. And when we stain the tissue, for example, we can see here in red that the Candida albicans that we put in is forming microcolony clusters. And these microcolony clusters are really invasive and inducing a lot of damage to the lung, not the lung epithelium, but the gut epithelium, and also to the endothelial side. And these microcolonies, or the hyphae consisting of these microcolonies, they are also able to translocate, as it was shown for the Aspergillus, from the intestinal to the vascular side. We also performed basically the same experiments by exposing antifungals within the vascular perfusion, as it was already shown for the Langenchip and Aspergillus fumigatus. So we again put in caspofungin, and now we expanded the study a bit more by not only including a wild-type strain of Candida albicans, which is known to be really susceptible against caspofungin, but we also inoculated a clinical isolate that was known to carry a resistance against caspofungin. And indeed, we can see here on the membrane images that the resistant clinical isolate, that there was no killing effect of the caspofungin, because there is still a lot of microcolonies forming after inoculation or infection of the model. And this we quantified with different parameters. This is only one snippet to showcase you how it basically looks like. So the intestinal fungal burden with the wild-type strain, here shown in red, decreases concentration dependently, whereas the resistant isolate, the intestinal fungal burden, remains very high. And intestinal fungal burden basically means we retrieve colony-forming units from both the tissue lysate, but also from the supernatants of the vascular medium, but also the epithelium medium. Okay, so that was it for the microbial infection part. I would now like to move on to the application 2. We're now expanding also our portfolio towards cancer-on-chip models, and our lead scientist, Dr. Tom Sommermann, did a great job in establishing this model here at Dynamic 42. We recently just published this model in Lab-on-Chip this year, and this chip has this chip model has a very unique feature, because we usually set it up in our biochip BC003, which is consisting not of 2 channels, but of 3 channels. So we have one top channel here, then we have a membrane interspan here, we have another membrane which is consisting of little microcavities, and then another channel here. So it's basically a 3-channel chip and 2 membranes integrated. And the microcavities that are set in these in the second membrane, they also build the basis to integrate full 3-dimensional spheroids. In this case, it was tumor spheroids from PDAC patients. So it's a pancreatic adenocarcinoma, very aggressive tumor. But basically, also the integration of whole organoids is possible in the chip. And usually, these organoids or spheroids, they need very controlled ECM environment, so meaning a gel or ECM coating, and usually a thick coating makes it impossible to perfuse the channel. So this is why we integrated a 3rd channel to feed in with the perfusion the spheroids from below. And we are also able to retract these spheroids from the chip, so we can collect the spheroids, stain them for immunofluorescent markers, for example, here shown Ki67, so it's a proliferation marker, so it's a highly proliferative tumor. And we also stain for alpha-SMA, because the spheroids consisting in the PDAC model, they're not only one cell type, but it's a co-culture from pancreatic tumor cells, but also tumor-associated fibroblasts. And these fibroblasts also produce a collagen surrounding, or a tumor-associated collagen shell around the tumor. All right, and this model can also be used for anti-tumor drug testing. So Tom basically did a lot of experiments with this. He used ZAHA, which is a HDAC inhibitor. It's also known under the name Virinostat, so it's an anti-cancer drug basically. And again, as a intravenous applied drug, we also try to mimic this by including this in the vascular perfusion. And what ZAHA basically did is that it dose-dependently decreased the cell viability of the spheroids. So after we extracted the spheroids, we can also do a lysate-based cell viability assay, for example, and quantify the viability of the spheroids. And this was reflected here. So ZAHA was basically really effective in killing the spheroids. But of course, we wanted to get sure that ZAHA is not only killing the spheroids and is relatively safe to the endothelial cells. And this is basically what is reflected here. So we quantified again the barrier integrity of the tissue, and we can see that SAHA does not affect the barrier integrity, which means that the endothelial cell lining is basically fine. So SAHA does not affect the cells in the endothelial compartment or the vascular compartment, but is effectively killing the tumor spheroids. Another cool feature is also the application for immuno-oncology. So the same model was basically used to integrate also circulating immune cells. So they circulated monocytes inside of the system. And these monocytes are really infiltrating the spheroids and also attaching to the spheroids. And they then polarize to macrophages if they are in contact with the spheroids. And what was really striking to see is that these monocytes, they really attach to the tumor as it's shown here. So these labeled green dots, these are the macrophages. And we were able to also fix these macrophages by extracting the tissue afterward. And these macrophages were shown to have an anti-inflammatory phenotype, which is shown here by the expression of this marker. So these are two anti-inflammatory markers, and these are two pro-inflammatory markers. These are go down, these two go up. And we can see that this is a very highly anti-inflammatory macrophage phenotype, which is known in PDAC that the macrophages or the immune cells themselves, they're suppressed during this kind of cancer. All right, moving on to our third application, the investigation of drug-induced liver toxicity. This is another study I basically did. The model was established in 2015 from our CEO, Dr. Knut Rennert. It basically consists of all the relevant cell types that are present in the human liver. So on the one side here, we integrate usually either hepatic cell line or primary hepatocytes, which are separated here from the vascular component, again, having liver sinusoidal endothelial cells, and also the immune component, which are macrophages in our case. Again, we were able to extract the tissue, to stain it for IF, to make beautiful images out of this, to characterize the model, whether these cells really express the cell markers they should have. They're cell-specific markers, definitely. And we're also able to perform enzymatic activities. So CYP3A4 is one of the main known drug metabolizing enzymes. And we were also able to demonstrate that with rifampicin, which is an inducer of this CYP3A4 activity, is really able to induce this activity. So we're really able to show this in our liver on-chip model. And we also compared primary hepatocytes compared to the hepar-G cell line, seeing that the primary hepatocytes, of course, they perform a much more physiological way compared to the hepar-G cells. All right, and then this model was basically used to test some drugs that were known to be withdrawn from the market and compare them with their structural analogs. And I will just showcase you one example, which is trovafloxacin. It's a broad-spectrum fluoroquinolone. This was withdrawn from the commercial markets after causing severe hepatotoxicity. And basically, the toxicity of this drug compound, it wasn't really seen in preclinical models, not in animal models, not in two-dimensional cultures. So we wanted to see whether our model was able to predict the toxicity of this drug and compare it also to levofloxacin, which is a non-toxic analog that is still on the pharmaceutical market. And we again did immunofluorescence staining, and we can beautifully demonstrate that trovafloxacin at human relevant concentrations really led to a destruction of the hepatic tissue and to a downregulation of the hepatocyte specific markers compared to the non-toxic structural drug levofloxacin. We were able to quantify the viability. We see a concentration-dependent decrease in cell viability when we applied trovafloxacin in vascular perfusion. However, this was not the case for levofloxacin. And also, we sampled the supernatant, as already introduced by Marty, and we were able to quantify the release of clinical tox parameters. Here, in this case, is ALT. And we can see a sharp incline in ALT release if the models were treated with trovafloxacin compared to its structural analog or the control models. I further performed then life cell imaging and see that also reactive oxygen species were drastically elevated in the trophofloxacin treated models, and that in the same context, glutathione levels, which are usually high enough to maintain cell viability here, they were also decreased upon hydrofloxacin exposure. Okay, these were now some examples. There are basically a lot of different things you can do with organ-on-chip, a lot of different organ models you can simulate. If you want to go for the lung, if you want to go for the liver, or develop maybe a brain organ-on-chip model, this is everything is possible basically. We are now focusing on different aspects. For example, on interconnecting also organ models. So we published this year a study interconnecting 3 different organ models. So we can just connect our chips together and perfuse the models like in parallel. So we published a study about gut, liver, placenta on chip model for drug distribution study. And this is basically how the whole model setup looks. Looks very complicated. Basically, a 3 organ model is quite complicated. But this is definitely something which is possible here to do at Dynamic 42, and this can also be implemented in the lab. And also 2 organ models, such as the gut-liver axis, for example, are possible, or the lung-liver model. Another very important feature for live monitoring of different kinds of readout is the integration of sensors or real-time measurements. On the left-hand side, you can see that we're currently working on the integration of tear electrodes, and this will most likely launch by the end of November. So we're integrating fixed electrodes inside of the chip, which can measure the barrier integrity of our models in real time. And it's also a pretty cool feature that you can put everything inside of the incubator and your laptop stays outside, because this device can act via Bluetooth. So you do not have to do anything. It just records the barrier integrity over time and measures barrier disruption, for example, if you put in a toxic agent. On the other hand, we already integrated successfully sensors for oxygen monitoring. So we integrate little sensor spots within our Dynamic 42 chip platform and combine it with a readout plate, which is shown on the right side. And therefore, we can measure oxygen levels also in real time, which is definitely important if you want to control the oxygen levels. For example, if you want to put in microbial species that are only known to grow under anaerobic conditions. And there are a few things that we're also currently working on. So we're developing an aerobic-anaerobic interface chip for improved microbial culture. We're having a project running regarding the establishment of a blood-brain barrier model. Of course, since cancer is a very hot topic nowadays, we're currently expanding our tumor on-chip portfolio, not only being restricted to the PDAC model, but also expanding to different areas of tumor biology, but also tumor oncology. We're not integrating only 2-dimensional vascular cell layers, but we're also working on the generation of 3-dimensional vascular networks, for example, also to vascularize our tumor on-chip models. And another very cool project, in my opinion, is also that we're working on the delivery of nanoparticles for antifungal treatments. And I mean, as I said already, there's so many options. Maybe we find yourself, or we find your application here in the next webinar. So there are a lot of different possibilities and opportunities for modeling different diseases or treatments or any other aspects. All right, so basically just a short summary how you can basically benefit from the Dynamic 42 models. So as I mentioned in the applications, you can model complex diseases or any kind of infection models. So bacterial, viral, or even fungal infection models are definitely integratable. Our models are completely immunocompetent, so we're at least having one immune cell type always present in our model. And we're generating only human relevant data because we're only using cells from humans. So we're not doing animal research here, but focusing on the data that we get from human cells. And integration of vascularization is also a key feature of our models and the biomechanical stimulation through the application of dynamic perfusion. As I said already, our models are customizable and scalable. So if you have a specific inquiry, just let us know. And not only our models are customizable, but also the chip platform itself is very scalable. So can be adapted to the need of any customer. And of course, as I mentioned already, just a little bit, working with human cells might be the more predictive model. So it may also lead to fewer animal studies and also to less paperwork and quicker results. All right, and with this, I would like to thank you very much for your attention. Feel free to use your phone to scan the barcodes for our website and our scientific references. And I would now go over to the questions that we got up front from the registrants. So I will just go through there one by one and try to answer them the best that I can. I think afterwards there is enough room to a lively discussion and Q&A. I think there might be a lot of different questions from the audience. All right, so let's get started with the questions. So the first one that we got is very basic question, but how can someone start their career with organ-on-chip with no prior experience? I gotta say, my experience is organ-on-chip technology sounds very complex and complicated, but basically it's not rocket science. So everyone that is a trained cell biologist or at least has some experience with pipetting and stuff can also use organ-on-chip technology. Basically, you would just need chips. You would need to sterilize the chips. You would need a coating of the membrane, and then you would need to seed in your cells and culture them under perfusion. So still, it sounds all very complex, but in the end, I would say it's not a big deal, to be honest, if you have previous laboratory experience. Of course, there's some tips and tricks, but you might learn this also if you join our on-site academy course, for example, or go online through the online academy. Okay, the second one is what particle size and solute content is optimal in air-liquid interface cell exposure? This one I found was a bit tricky, so I had to look a bit up because I'm not a lung biologist specialist, but I also got back to our scientific lung-on-chip team, and they suggested that particle size should be between 100 nanometers and 3 micron. And the solute content should be below 1%, so there should not be a lot of salts or ions in there, because during the aerosol exposure it can evaporate, and then you get crystals inside of your model, and this is something you do not want. What are the benefits and challenges of liver-on-chip? So the benefit is definitely that you can do a very nice in vivo-like drug testing, for example, putting in your drug treatments during or within the vasculature. So this is really mimicking a human intravenous administration route. And for the model itself, it benefits a lot from the perfusion, because there are a lot of studies out there showing the benefits of perfusion in the context that the metabolic activity is increased. Also, the longevity of the hepatocytes is increased. So the model itself really benefits also from the perfusion. And in comparison to animal models, it's a lot faster, less bureaucracy, and cost-wise is also better. And challenges, I would say that the cell sources, particularly the hepatocytes, they differ very much from batch to batch. So you have to test out a lot to find a really reliable and good batch of the cells. This accounts basically for all primary cells. So this is something you got to test out which cell type or cell source or cell batch works best for you. And challenges would also be the upscaling, maybe the throughput of the model. So speaking about general screening of drug toxicity in the liver-on-chip model needs to be expanded a bit more, since we are only addressing low to mid throughput. High throughput is definitely a challenge that is ongoing in the liver-on-chip. What is your intake in using this organ-on-chip as a skin and dermal model? To be honest, for topical administration of compounds to see topical toxicity, I would always recommend to 3D constructed skin tissues, because they are already very in vivo-like. But I would recommend organ-on-chip in the context of intercellular interactions. For example, if you have a scientific question regarding skin and immune cell interactions, or if you want to have a systemic distribution, for example, of your compound. So you administer a compound on top of the skin, so topical administration, and you want to know how much of the compound is found, for example, in the liver. You can also use a skin liver, for example, multi-organ on chip. So you always have to adapt the model a bit to your scientific question or your experimental need. But in the end, this is something possible. We are not at the stage where we provide a skin-on-chip model here at Dynamic 42, but maybe this is something that could be developed in the future. So the next one is best way to start growing cells on both sides of the membrane. I have the feeling that it's that the question is in that direction, how you put in cells on both sides on the membrane, because otherwise they would just fall down if you see them somewhere. So basically, on the membrane, you definitely need a coating, so the cells really have a good scaffold where they can attach on the membrane. Some cells also attach on the membrane without any kind of coating. And usually, if you want to seed cells on the top, you just seed them in with a pipette in the chip, and they will sediment on the membrane. But in the bot channel, it's a bit tricky. So if you have seeded the cells on the top, you would seed in the cells on the bottom channel, but you need to turn the chip upside down. So the cells are not falling down on the foil of the chip, but if you turn them upside down, they will also sediment on the membrane. And then you have really two cell layers on both sides of the membrane. Okay, the next question would be lung, skin, and gut MPS with and without immune components. Not quite that if I have the correct answer for that, but basically we do lung and gut MPS here, Dynamic 42. Skin might be worth the development, and with and without immune components, I think I said this quite a lot during my talk, that we're trying to work immunocompetent, meaning that there will always be an immune component within our model. Okay, we have 3 more to go. So the problems in organ-on-chip technology and suitability and sustainability for drug studies. So this is, I think, a bit in relation to the third at the beginning. So the benefits and challenges of the liver chip. The problems in organ-on-chip technology, I would say, are still the broad adoption, so the regulatory gaps that are still ongoing. So the FDA has now announced to do a lot in this field and also push the field of organ-on-chip. But I think still there's no organ-on-chip model that is currently approved during the regulatory authorities. So a widespread adoption is definitely nothing that is currently ongoing. And there are some platforms that can do high throughput, but I would believe that most of the organ-on-chips, they're still limited to low or up to mid throughput. Other problems might also be the availability of the cell material you put in the chips, and also speaking about the material, most organ-on-chip vendors, they might use PDMS, which can be a limitation regarding small molecule studies, because the small molecules, they bind a lot to the chip material, so the PDMS. So this is basically why we're using a non-PDMS chip. So it's a material that we're using that is low in absorption of different drug compounds. Yeah, I think that's basically it for the drug studies. I think I've already answered quite a bit. So let's move on. Is the pump used in Dynamic 42 biochip specially designed? Not specially designed in that way, because it was already designed like this, but we chose a pump that fits our need, I would say. So the pump head itself goes in the incubator, so you can directly work inside of the incubator. You can put your chips on the pump directly in the incubator, and every unnecessary electronics, for example, the external controller of the pump head, that goes outside of the incubator. So we have both the pump head in and the controller outside of the incubator. And the pump heads are also designed to stay for a long time in the incubator. So usually we have a good warranty on that the high humidity inside of the incubator is not interfering with the work of the pump head. And the last question is the inclusion of ECM matrices in organ-on-chips. So I can that for all our applications, we use ECM in different kinds of and concentrations. So you can either do a thin layer, for example, a top layer or minion layer, but you to include thick gels for a 3-channel chip. If you want to integrate spheroids or organoids, then you can also put in, for example, matrigel or other kinds of gel which are really thick, and put the cells in their 3-dimensional arrangement. Yeah, I think I answered all the questions that were already put up front, and I think Jule, you already collected some more, right?
Some more is an understatement.
Okay.
I think we've got like 30 or so.
Whoa, that's a lot.
So, yes, I hope we will get through them with the time we've left. If we don't, I will already say apologize, but we will answer them directly to you afterwards in emails, so don't worry about it. So just before I forget, I will launch a survey just to ask you how you like the webinar. And while you're answering this, I will read the first question to our guys, Tim and Marty. So the first one is, how does the microimaging of the chips, or does the microimaging of the chips require specific lenses?
Mm-hmm. So maybe I can take that over. So I'm not quite sure what microimaging is. I'm only familiar with fluorescence microscopy, so or light microscopy. Usually for microscopy, you can either extract the membrane from the chip, so just cut it out with a sharp scalpel and then stain it for different markers of interest. Or what you can also do, as said, you can do live imaging, so you can put the chip directly under the microscope. Regarding microimaging, I'm very sorry if I'm not familiar with that, but I have to pass for that. Sorry.
Okay. So next question is, in the case of lung-on-chip, can we perfuse the lower chamber, so the endothelial side, with a solution containing neutrophils? If yes, the cells would be able to cross the endothelia and the AEC barrier during the infection?
Mm-hmm. Also very good point. So what we also do is integrating circulatory immune cells, meaning also neutrophils. So either you get neutrophils from a commercial vendor already isolated, or I think you can also isolate neutrophils from a human PBMC, so from donor blood, for example. Putting them in the lung chip is definitely possible, so you can integrate them just in the circular perfusion, and then it depends a bit on the microbial strain that you put in. So if it's really a severe infection, I believe the neutrophils will also react accordingly and also secrete a lot of cytokines, and might also be able to migrate through the membrane. Maybe in this regard, so we offer different kind of pore sizes for the membrane. For transmigratory studies, such as your application, I would then recommend choosing the 8 micron or 5 micron. We also have a 0.4 version where basically no cells can migrate through. But for this transmigratory studies, I would definitely go with larger pores, because the cells are able to migrate.
Thank you. So here are two. I have a few questions, here two of them. Are your chips capable of applying mechanical stimulation? That'd be the first.
So basically mechanical stimulation, meaning I think stretch and strain. This is something we cannot really integrate in our system because the chip is relatively rigid. So this is, unfortunately a downside of the material that we are using. For drug studies, it's very useful, but for stretch and strain, not really. We have the biomechanical stimulation by the perfusion itself. So this is one of the biomechanical stimulations that is possible. And we are also working on different options for the lung-on-chip model to also deflect the membrane. So this is something we're trying to implement and integrate, and this will hopefully also come soon.
And the second one's about your lung-on-chip device. You mentioned that you're able to culture both alveolar and bronchial cells. But these cells cultures don't translate to an appropriate mimicking because the alveolar architecture is completely different than the bronchial. Hence, you need two different designs for each part, alveolar and bronchial. How much does your design respect the 3D structure of alveolar sacs?
Yeah, I mean, this is maybe also another downside of organ-on-chip organ modeling. I mean, speaking, you can model some subunits of the organ model, but recapitulating a whole organ model, of course, is something that is maybe an illusion and will never be achieved. We're trying to set up some small functional units of organ models and putting in the key features of the cell types or the relevant cell types that are really necessary to perform biological functions. For the alveolus, I can also get you in contact with our lead scientist. I'm not quite sure about the alveolar biology in very much detail. But I would say for all our organ models, we keep to the small functional unit of the organ model.
Thanks. So then another question, microscopy could be a wide field microscope used to image chips.
Yeah, this is definitely possible. So usually we're just taking a chip outside of the incubator, put it under the microscope, and through to the transparency of the bonding foil, it's relatively easy to assess this. Yeah.
And then another one is during the development of co-infection, bacteria and virus approach, how does your team account for key physiological functions in vitro, such as cell cycle regulation, tissue injury and repair, and disease progression?
So basically, the study performed a lot of different assays. So usually we're using our standard panels such as barrier function, staining of morphological features. We're also studying transmigration of the microbes by just plating the supernatants and checking for the CFUs. So these are like the things that we are regularly doing. Of course, there are always more biological assays that you can do with, for example, cell cycle progression. I think you got to check that with other biological assays. This is just what we did, but basically you're working with the chips, you're not really limited to other kinds of readouts. So if you have a specific application or readout that you want us to be covered, then just please let us know.
So also regarding variability and reproducibility,
how do factors like cell source selection, similar to Sabina's, I get a question before about cell line selection, chip design or material choice impact the standardization and consistency of the results?
Yeah, it affects them a lot. So in regard to the material, I mean, this is something you do not regularly change, I would say. So if you found a really good material that suits your need, then I think you're good to go. Depending on the cell sources and cell lines, I think it's a bit of a different story. So every cell batch can react in another way. So for real standardization and reproducibility, it's often really advisable to cross-test also different batches, but also different kinds of cells. So usually a comparison between cell lines and primary cells or even IPS cells is desirable when it comes to really reproducing effects, for example, for toxicity testing. So yeah, but these are just two aspects. So this is a shortcoming, I would say, of organ-on-chip, that there are a lot of different parameters that you have to keep in mind, and it's relatively hard to standardize them. But I think they're particularly in the Eurox consortium, for example, or the CN and CN LEC group in Europe. They're currently working also on the standardization of these models. So hopefully they I think they have published a roadmap already this year or last year, so showing a lot of parameters. So if you're interested, you can also have a look in there.
So now some more sort of the lab
Work itself. How do you maintain sterility of the chip? So how is it incubated? How do you actually change the medium? The membranes are integrated from the beginning, or should we play them based on the cell line we need?
So very good question. So this comes up a lot of times during some webinars or customer inquiries. So basically the chips, once they're at perfusion, or if we seed in the cells, usually we try to keep them as tightly sealed as possible, that there's no room for contamination or sterility issues. Usually we start, as I said, by sterilizing the chips with ethanol. So there's no risk of contamination there if you want to seed in your cells afterwards. For the culture of the model, it's everything very, very leak-proof and tightly sealed. So we don't have contamination issues at all here. So this is working very well. Yeah.
And now to cells. So instead of immortalized cell lines, is there a way to use iPSCs from patients carrying specific mutations to establish a patient-specific chip?
Yeah, definitely. So we also have collaborators here in Germany, but also in other countries using iPSC-derived cells on the chip. So as I said, you're not really limited in the cell source you're having available. It just needs a bit of fine-tuning what kind of a matrix or ECM these cells are needing in the chip and what kind of medium you use to perfuse them or you culture them under static conditions. So if you're already having your cell types or cell sources in your lab and you know how to handle the cells, I think there are not many limitations to integrate them also in a chip-based model.
Cool. And then again on reactions, are cells resuspended in any matrix or just injected in the channels when assembling them and waiting for them to attach to the bottom? Is there a vascularized compartment or do you have vessels? When you co-culture cells of a certain niche, or do you have different medias for each channel?
Okay, I try to keep that in mind. So first one, whether we're going to see it in the cells in a gel or not, or in an ECM. Basically, so what we do is to just pipe it in the cells in suspension, so in medium in the chip, and let them sediment on the membrane. So usually they attach very well. It's also possible to directly seed them in a gel. So for example, such as a matrigel. So we already did it for hepatocytes, for example. Another part of the question was if I reckon it right to that we can have different kinds of medium in each compartment or each channel. This is definitely possible as long as your cells are growing confluent and the barrier is tight. So you can put in one medium in the top channel, perfusing your endothelial cells, and can use epithelial-specific medium in the other one. Definitely possible. And in the middle, there was the thing with the three-dimensional vessels. So I got everything. With the three-dimensional vessels, our lead scientist, Tom Sommermann, is currently working a lot with these. So we generated really nice looking three-dimensional vessels, also putting in beads where you can actually perfuse beads through these vessels. So they really have an enclosed diameter. But this is ongoing work. If you're interested, just shoot us an email or an inquiry and we can talk more about that.
Okay. Another one. Can you mimic hypoxia or anaerobic environments of the lumen for garden chip model?
Very good question. This is also ongoing work. So basically, we are also in a collaborating project together with other partners where we are establishing this. So this is gonna come most likely beginning of next year, maybe. Let's see how far we progress. But this is something we definitely want to integrate, especially if you're looking for anaerobic culture or even microbiome culture within the gut on chip. Yeah.
And then one to the upcoming tear sensors. What tear values do you obtain for gut on chip? So ohm per square centimeters?
Yeah, also a good question. So usually we set the tear values in relation to the culture area, so the membrane area basically. And this is basically what we do. We also provide the tear values in ohm per square centimeter of membrane area where the cells sit. Usually the values under perfusion of the gut model are usually between 200 to 400, usually in the 300 range. Yeah.
Cool. Now one that I feel like I could even answer that, but is the plastic
window... Go ahead.
I'm gonna try. Is the plastic window not a problem for in situ imaging? Could you improve it in the future using glass? And I've heard we're doing custom chips that do have glass instead of the plastic, but I'm going to leave it still to.
Really? Okay. Oh, I would have taken the break. No worries. No, definitely. So as you already mentioned, we're also providing chip with glass bottom instead of the foil. Usually my experiences with the foil already works very well with fluorescence microscopy, also confocal imaging. With the glass, you have a better refractory index. So they are a bit more suitable for imaging, but if you extract the membrane anyways and put them between two glass slides, basically you have glass. So it's more useful for live imaging in the chip, that you put the glass bottoms or glass ceiling instead of the falls. But if you're not wanted to get that one, just you can extract the membrane and stain it and then put it between two glass slides.
Cool. Yeah, that was a little more comprehensive than my answer. Okay.
Mucosal macrophages are conditioned by the immune environment and metabolites. So the model might not predict tolerogenic responses like in the lung or the gut.
Okay. Yeah, very difficult question. To be honest, I would say the immune system is so diverse that, of course, integrating only one immune cell type might not reflect every kind of immune reactivity that's happening in the body, especially not when it comes to tolerogenic responses. So I have to say it's quite a limited immune system. It's not a fully one. You can add, for example, circulating PBMCs. There you have basically a lot of different kinds of immune cells, T cells and other kinds of immune cells. And so expanding this is definitely a good thing when it comes to adverse events or tolerogenic responses.
Are the cells from different membranes growing into mixed clusters?
Or they are kept within their physical space?
Can you repeat that one, please?
Sure. Are the cells from the different membranes growing into mixed clusters, or they are kept within their physical space?
This basically depends a bit on the membrane itself. So whether you have a plain membrane or you want to have the membrane with a microcavity. So with the microcavities, you already have a capsule structure where the cells can be arranged in a three-dimensional arrangement or morphology. And if you have the integrated plain membrane without the microcavities, the cells most likely will grow as a confluent monolayer and remain in their configuration. But if you want to keep your spheroids or organoids, for example, in a three-dimensional morphology and you do not want to fragment them or disrupt them prior to seeding, then integrating them in the cavities, in the microcavities, would be a good choice. I hope that was the right answer. If not, I'm very sorry.
Okay. So multi-chip connections will open the door for lots of studies.
But this may also require multiple peristaltic pumps and a huge space in the incubator. Do you have suggestions on pumping systems?
Also, very good remarks. So, of course, we're always looking for new pump systems, especially when it comes to micro pumps. Scaling up is always a thing that needs to be addressed in the future regarding organ-on-chip technology. But I mean, you have seen it in the incubator. There were like 2 of the 3 organ models. If you want to upscale this, you need either more incubators and more pumps, or you just reduce everything a bit. So this is a technical advancement that we're currently working on, to trying to get rid of larger pumps and moving also to smaller pump systems, for sure. Yeah, good remark.
Okay, seeing that we've already went past 5, let's say we do 2 more questions.
Fair enough.
And then
I will close the webinar also to give Tim a bit of a break.
All good.
Okay, so here they come. So second last, how can you make a liver on chip that works for more than 2 weeks if hepatocytes or primary cells do not survive more? Or does vascularity, so the vasculature and the whole 3D system maintain them longer?
Yeah, so basically the perfusion and also the intercellular communications between endothelial cells and hepatocytes, they are known to induce a longer lifespan of the hepatocytes. Longer than 2 weeks is still very long for a primary model, depending on the cell batch you have from the primary hepatocytes. Usually they're very picky. There are different ways in expanding this. For example, overlaying the hepatocytes with another matrixal layer, so having a sandwich culture of the hepatocytes is something that can be achieved in the chip and can also increase the longevity of the hepatocytes.
Okay, super. And now the last one. How do you deal with DMSO interference in interpreting transcriptomic
on HEPA-RG DMSO treated?
So usually for DMSO, so the HEPA-RG culture usually requires 2% DMSO for letting them differentiate. Transcriptomic changes I cannot really tell much about because I haven't checked it for our HEPA-RG culture. But I can tell you that if you move into the chip, we usually reduce the amount of DMSO for the HEPA-RG to 0.1%, so it doesn't have that much of an impact. This is also mainly due to the fact that DMSO is toxic to other cell types, such as endothelial cells and also immune cells. So this is a main reason why we reduce the DMSO. Speaking about transcriptomics and the influence of DMSO, I'm sorry, I have to pass about that.
Okay, so thanks so much, Tim, for answering all the questions.
Okay, so with this, I'd like to end the webinar. Thank you all so much for joining us. Like I said, I was in the recording, and I hope you join us again in the future. Thank you. Bye.
Thank you for joining.